What if gene editing is moving from fixing typos in DNA to replacing whole chapters? That sounds dramatic, but it is exactly the metaphor researchers used for a new technology reported in Nature on April 29, 2026. The method is called prime assembly, and the idea is simple to say, but hard to do. Instead of changing one tiny DNA letter, insert a large, healthy DNA segment directly into the genome. For gene therapy, that difference matters. Many genetic diseases are not caused by one neat mutation that every patient shares. Some genes can contain hundreds or even thousands of different disease-causing mutations across different people. If you try to fix every mutation one by one, the problem becomes messy, fast. But if you could replace the broken version of the gene with a healthy copy, you might not need a custom edit for every possible mutation. That is the promise behind this new work. The researchers built on prime editing, a newer gene editing approach designed to make precise changes without cutting both strands of DNA in the classic way. Prime assembly goes bigger. It uses a strategy involving overlapping DNA flaps that help a donor DNA segment connect with the target genome. The important part is that the donor DNA does not need a double-strand break, which can be toxic and can kill cells. In mammalian cell experiments, the team reported insertion of DNA segments up to 11,000 base pairs long. For comparison, the article notes that some other methods had successfully inserted around 800 base pairs. That jump is why this is interesting. A few hundred base pairs is like editing a sentence. 11,000 base pairs starts to look more like replacing a full section of the instruction manual. One of the lead researchers described it this way: if the genome is a book, this method could remove a paragraph and replace it with a new one, or even rewrite a chapter. This is not just a nicer metaphor. It changes how people imagine future therapies. Take a disease where patients have many different mutations in the same gene. Instead of asking, which exact mutation does this person have, a future therapy might ask, can we install a working copy of the gene? That could be especially important for diseases where thousands of rare variants exist and each one is too uncommon to justify a separate custom therapy. But there is a big reality check. This is not a cure you can get next year. The study shows promise in cell cultures. The next challenges are delivery, safety, efficiency, and proving that the system works inside living organisms. Getting a large DNA segment and the editing machinery into the right cells is one of the hardest parts of gene therapy. Possible delivery vehicles include lipid nanoparticles or adeno-associated viruses, but each option has limits: cargo size, tissue targeting, immune reactions, and manufacturing. There is also the question of control. When you edit a genome, precision is everything. Scientists have to measure not only whether the right DNA was inserted, but whether anything unintended happened elsewhere. That is why the cautious interpretation is the correct one. Prime assembly is not magic. It is not a universal fix for every genetic disease. But it does suggest a different direction. Not just snipping DNA, not just changing letters, but assembling larger pieces of genetic information inside cells. If that direction works, the long-term impact could be enormous. Future medicine might treat some genetic disorders not by chasing every mutation, but by replacing the damaged instruction with a clean version. That would shift gene therapy from correction to reconstruction. And it shows how quickly the language of biology is changing. For decades, DNA was described as a code. Then, gene editing made it feel like text that could be corrected. Now researchers are trying to move beyond fixing typos. They are asking whether we can rewrite whole chapters. That is why this breakthrough matters, not because it is ready for patients today, but because it expands what genome editing might eventually become.